Hello, I'm Henecy Marquez. Today, we will discuss about the mutation and gene regulation. Mutation. A mutation is a permanent change in the DNA sequence of a gene. Mutations is a gene's DNA sequence can alter the amino acid sequence of the protein encoded by the gene. The substitution mutation, when a nucleotide in a gene is copied incorrectly during DNA replication, one nucleotide can be substituted with another. This results in the incorrect amino acid sequence, changing the structure of protein. Here are the correct DNA sequence, the correct RNA transcript, the correct polypeptide. Substitution mutation, resulting transcript, resulting polypeptide. Silent mutation. A mutation is silent if it has no effect on the protein coded by gene. The reboundary in the genetic codes make this possible. This reduces possibility of a mutation located in a gene causing a change in the protein. ALA, RAVARG. Each amino acid has more than one possible codons, so if a substitution occurs, the same amino acid may still be coded. Mutagens. A mutagen is a physical or chemical agent that can change the DNA of an organism and thus increases the frequency of mutation. Mutation include ionizing radiation such as X-rays and gamma rays, ultraviolet waves such as sunlight, mild, alkaloid plants such as tobacco, coca plant, and poppy plant, sodium acide such as component in many car airbags, benzene such as solvents used in plastic synthetic rubber. Spontaneous mutation. Not all mutations are caused by mutagens. Spontaneous mutation occurs due to error involving changes to chemistry of the DNA, DNA replication, repair, and recombination. So, are mutation always bad? Well, not necessarily, very rarely. A mutation will cause an individual to become stronger than the rest of its population. Sometimes, a mutation can give a prokaryotic resistance to no antibiotic or toxin. Now, let's go to gene regulation. Gene regulation refers to the mechanism that act to include or repress the expression of a gene. These include structural and chemical changes to the genetic materials, bending of proteins to specific DNA elements to regulate transcription, or mechanism that modulate translation of mRNA. Regulatory protein positive transcription factor, RNA polymerase, regulatory protein negative transcription factor. RNA polymerase. RNA polymerase is an enzyme that synthesizes RNA from a DNA template. Using the enzyme's helicase, RNA locally open the double-stranded DNA so that one strand of the exposed nucleotides can be used as a template for the synthesis of RNA, a process called transcription. Prokaryotes. A prokaryote is a cellular organism that lacks an envelope enclosed nucleus. The word prokaryote comes from the Greek "epo" and "kapo" in the two empire system, arising from the work of Edward Chatton. Prokaryotes were classified within the empire Prokaryota. Some eukaryotes called the operand. So what do we mean by operon? The operon model of prokaryote gene regulate was proposed by Marquez, Jacob, and Jacques Monod. Groups of gene coding for related protein are arranged in unit known as operon. An operon consists of an operator, promoter, regulatory, and structural genes. If the repressor protein is removed, transcription may occur. Enzymes. The enzymes genes that codes for amino acid sequence are known as structural genes. The process of gene expression involves two main stages. Transcription, the production of messenger RNA, mRNA by the enzymes. RNA polymerase in the processes of the resulting mRNA molecule. Transcription and translation are the step of protein synthesis. One is promoter. Promoter is a sequence of DNA needed to turn a gene on or off. The process of transcription is initiated at the promoter, usually found near the beginning of a gene. The promoter has a binding site for the enzymes used to make a messenger RNA, mRNA molecule. Second one is the operator. An operator is a genetic sequence which allows protein responsible for transcription to attach to the DNA sequence. The gene or genes which get transcribed when the operator is bound are known as the operon. The function of the operator within genetics is to regulate the production of a certain portion of the DNA. Last is the repressor. A repressor is a protein that turns off the expression of one or more gene. The repressor protein works by bending the gene promoter region, preventing the production of messenger RNA, mRNA. And the lac operon. The lactose operon or lac operon is an operon required for the transport and metabolism of lactose in E. coli and many other enteric bacteria. Notice that lac operon is promoter DNA operator region of the DNA and the LACZ, LACY, LACA. Lactose utilization genes. These genes codes for three different enzymes that have very different roles in the utilization lactose. That's all. Thank you and God bless.